There's been a data explosion. If you think about the number of devices in the world today, every single device is generating more and more data. And I think we all know that that's not going to stop.
Welcome to Hidden in Plain Sight. I'm Sophia Bush.
And I'm Austin Craig.
This podcast explores the real-life people, teams, and technologies that are solving society's most serious problems.
Right now, all across the globe, there are a handful of people crazy enough to believe that they can solve our biggest dilemmas with data.
If we're going to find solutions, we have to know where to look. And as more and more of our lives are digitized, even more of those answers can be found in the data all around us. What if the answers to humanity's biggest problems were hidden in plain sight? This season of Hidden in Plain Sight is brought to you exclusively by our friends at Splunk, the Data to Everything platform. Splunk helps organizations worldwide turn data into doing. It's time for data to be more than a record of what happened. It's time to make things happen. Learn more at Splunk.com or by clicking the link in our show notes.
In 1928, Alexander Fleming was studying cultures of Staphylococcus, a bacterium that causes boils and sore throats, or what we would call a staph infection. But while Fleming was on vacation, one of his petri dishes got moldy. As he was about to throw out the dish and the bacteria, he noticed something strange. Right around the mold, in a broad perimeter that should have been filled with bacteria, there was nothing. The mold had somehow prevented the bacteria from spreading. Fleming had accidentally discovered penicillin, and by extension, the entire category of antibiotics. Science author Steven Pinker estimates penicillin alone has saved roughly 82 million lives, not to mention the dozens of other antibiotics developed since. But this was an accident. What if Fleming had thrown away the moldy petri dish? Happy accidents are great, but they're not a reliable means of discovering medicine. And sadly, in the past 90 years, the process of developing new drugs hasn't improved as much as you might think.
It's simply not a sustainable system where we can come up with drugs for all of these patients in a time and at a cost that's actually feasible.
This is Ginny Deshpande, the founder and CEO of NewMedi, a Silicon Valley startup that uses big data and artificial intelligence to develop new drugs faster. The promise of artificial intelligence and machine learning is that one day, hopefully sooner than expected, Ginny and her team at NewMedi may be able to use data to quickly discover life-saving drugs and drug therapies. To date, there are more than 115 companies active in this space, tackling the drug discovery problem from various angles. One of those companies is Recursion Pharmaceutical, which uses robots to conduct experiments. Recursion co-founder and CEO Chris Gibson says that his team is generating 70 terabytes of data every week.
Now, 70 terabytes is a lot of data, much more than most people ever deal with. For perspective, in 70 terabytes, you could store 24 million songs, or half of Spotify's entire global library of music. You could store a standard definition copy of every movie Hollywood has made for the past 50 years, twice over. Or if you wanted to fill 70 terabytes with text, something like Webster's English Dictionary, you'd have to store 7 million copies to use up all that space. That's how much data Chris and his team generate every single week.
That's way too much data for humans to process, and humans are full of biases. And so we use computer vision and machine learning to analyze all of this data every week to help us identify opportunities where some interesting biological or chemical set is coming together, and these might be new treatments for patients.
Chris and his team begin in the lab by adding disease cells, for example, malaria parasitic cells, to healthy human cells. They let the cells grow in individual cultures, and throughout the week, introduce tens of thousands of different chemical compounds. At the same time, a microscope captures more than 10 million images. Then a computer vision technology, similar to facial recognition, identifies all the ways that those cells have changed. Algorithms track how the cell changes visually in shape, color, and composition. By automating this process, they can test in days what previously took years.
If a disease makes cells look different in some specific way, is there some drug that makes the cells look normal again? It's as simple as that. So we do this massive quantity of experiments using robots, and when we find this intersection of some drug or genetic pathway that intersect in some way using images, we then have a whole team who will dive in with a lot more rigor to try and understand that interaction. The value of every experiment we do should, on average, increase over time as our algorithms get better at predicting what drugs could be useful in each disease.
Imagine how long it would take for a team of humans to run these experiments without technology. Robots, as Chris says, don't get tired. In addition to data created in labs like Recursion, companies in the drug discovery space are helping make sense of other types of data that are harder to parse, like data that's collected by academic medical centers, pharmaceutical companies, and health data that's collected by patients independently.
So we're at this sort of very interesting cusp in our field of having lots and lots of data being collected across the ecosystem. And that's where, we have the ability to mine all this data and find connections that are hidden amongst this data. So for instance, people have been studying a certain field for 30 years. They've published lots and lots of papers in that field. What if you could apply something like AI technology to mine all of those papers? That's just one example of how technology can mine literature and find things that might not have been obvious to humans.
Ginny and her team at NewMedi are working with billions and trillions of data points, literally zettabytes of data. However, it's nothing compared to how much data other companies accumulate.
The data that we're collecting is still smaller than something like a Facebook, right? Facebook's collecting every aspect of your life. We're still not at that scale in terms of biological data that's being collected. It's at a much smaller scale, but it's getting deeper and richer in terms of us being able to mine it and make sense of it.
Making sense of that data requires that it be extremely orderly and formatted in a usable way. If the measurement units are not consistent, if measurements aren't collected the same way, it can throw off correlation or insight that might otherwise be found. It's what scientists call cleaning the data.
About 80 to 90% of the work that data scientists do today involves cleaning up data. And then the rest, maybe 10 to 20% of it is actually computing things with the data. So we're, making sense of it and coming up with new and interesting findings. Cleaning up the data takes a lot of effort. Some of it is still very manual. You have to understand context. Did this data set come from someone with stage 2 or stage 3 breast cancer? What drugs were they on, as an example? And being able to understand, was this from a biopsy? Was this from blood? Those sorts of levels of detail become really, really critical. So there's a lot of cleaning up of data that needs to happen.
This new industry is advancing, and there are still plenty of challenges to overcome. For one, there's a lack of training data to bring machines up to speed. When you're working with technologies like artificial intelligence, you have to train the machine to recognize patterns. But to do that, you need enough already identified patterns to effectively train the system. This scarcity of training data is currently a focus at Lawrence Livermore National Laboratories. They're partnering with the National Cancer Institute and the University of California at San Francisco to solve this. Here's Bill Goldstein, director of Lawrence Livermore National Laboratories.
One of the things that we're able to do by combining machine learning with the ability to simulate, for example, the docking of molecules with cells or with targets, is that we can actually add simulation data to real data, to experimental data, and greatly expand the amount of training data that's available to our machine learning systems. We can actually also guide the kinds of experiments that can be done in the future and optimize them to fill in the gaps that exist in the experimental databases. The computers that we have today turns out to be ideally suited to combining machine learning with actual physics simulation, biological, chemical, and physical simulation of systems.
Accurately simulating complex systems and experiments is one of the first steps toward combating the diseases for which there are currently no cures. But before we can run these simulations, we need to access all the different data that are currently siloed.
So for instance, we have data that's been collected in academic medical centers, for instance. So they may have taken biopsies from cancer patients and banked them. they collect these samples and keep them. So that's one silo of data. And then you may be able to get the profile, but you don't know what else this patient has, what else has happened to this patient. So for instance, you might get all the molecular profiles, but you may not know, things like what drugs were they on? Did those drugs work? Did they try multiple drugs or were they only on one drug? Things like that, which is the clinical data, the clinical trial data, or the electronic medical record data, may not be available.
Another challenge is that machine learning algorithms lack judgment. You can't put information into a computer and have the computer spit out a new drug to go to market. Human inquiry and curiosity lead the way. Machine learning just helps.
You still need to determine what data gets fed into the computer. That's a human process. You need to look at the output from the computer. That's still a human process. And then once you get your drug candidates from your computer, testing them in cell lines, in animal models is still very much the process. You cannot skip that process. The FDA requires you to go through that process.
Using animal models in the testing phase of drugs has always presented a constraint to truly understanding how a drug might interact with human biology. A human being and a mouse, for example, are quite removed from each other in respect to their evolution. But Ginny believes that technology can help here as well.
Imagine a scenario where you have, every strain of mouse model that's ever been looked at for a particular disease. Imagine if you could take data from all of these models and then match that to the human patients and say, how closely does this actually match? What aspects of it does this animal actually match for the human condition? So that when you test your drug, if it doesn't work, is it because the same mechanism doesn't work in the animal or doesn't exist in the animal? Or is it because there's something different there, right? So you can really start to get smarter about how we utilize the artificial systems that we have to go through in terms of testing our drugs. I actually see that as being sort of the next phase of growth of our industry. It's going to be needed.
It's still early days for AI and machine learning in the drug discovery field, but it's a maturing space that will see huge advancements in the next decade. Currently, there's no drug on the market approved by the FDA that's come from machine learning. But given the past challenges of discovering new drugs, the cost and time and even randomness of, say, finding penicillin in a moldy petri dish, Chris Gibson at Recursion is extremely optimistic about future results.
The machine that we've built is truly going to enable the discovery of new medicines at a scale that heretofore has been essentially incomprehensible.
Recursion is running 500,000 experiments a week. So it's not unrealistic to think that in the next 5 to 10 years, we could see a success story for the industry. And Ginny says that in the next 2 to 3 decades, these kinds of AI-discovered drugs will be the norm.
I think we will start to see this convergence of what patients are doing, collecting all this information about themselves, and I sort of see a continuous feedback loop between the next generation pharma companies and what patients are doing. There'll be a tight interaction between the patients and the companies, because it'll be a dynamic relationship where there's data flowing back and forth between each of them. There's going to be a lot of information exchange that'll happen. And so I'm actually hugely optimistic about the future.
There are more potential drug-like molecules than there are atoms in the universe. It's something like 10 to the 61st possible chemical compounds. If you extend that into biologics, this is essentially an infinite space and an infinite problem.
An infinite space and an infinite problem. Hopefully in time there will be finite solutions in the form of drugs and drug therapies. Until that day, Ginny, Chris, Bill, researchers, and robots are working hard to uncover the solutions in the data that are hidden in plain sight. I'm Sophia Bush, and you've been listening to Hidden in Plain Sight from Mission.org. This podcast is sponsored by our friends at Splunk, the Data to Everything platform. In today's data-driven world, every company, big or small, new or old, is sitting on terabytes of unused, untapped, and unknown data. Splunk helps turn all that data into action. Using cutting-edge AI and machine learning, Splunk delivers real-time predictive insights that will help you on your mission to change the world. With solutions for IT, security, Internet of Things, and business operations, Splunk empowers people to make faster, better decisions and take action to get things done. It's time for our data to be more than a record of what happened. It's time to make things happen. Check it out at Splunk.com.